are going to talk about enzymes. For the most part, enzymes are protein. There are some enzymes that are called ribozymes that are made out of RNA, but we are going to be focusing mostly on protein enzymes. Enzymes tend to end in ASE. Not all enzymes do. There are many, trypsin, pepsin, that end in other endings, but if you see something that ends in ASE, you should think enzyme. Enzymes are biological catalysts that help speed up the reaction rate. If this is an enzyme, the reactant to the enzyme is called the substrate. So the substrate is whatever is doing the reacting in an enzyme catalyzed reaction. Now, the substrate doesn't necessarily have to be one molecule. It can be a couple molecules that are going to be put together, but it is going to be complementary in shape to the enzyme. So where it sits on the enzyme is called the active site. So this area here of the enzyme where the substrate is going to sit and react is called the active site. So the active site is complementary to the structure of the substrate. This is going to make the enzyme specific. So enzymes are specific to the reaction that they are catalyzing. So the substrate will sit on the active site, the reaction will occur, and then the product will leave. Enzymes can be reused as long as they are active and not blocked. There are a couple different ways that organisms can block the enzyme from being utilized. And we'll talk about that in just a few. Enzymes lower the activation energy of a reaction. If we are starting a reaction that is going to be catalyzed by a particular enzyme, and looking at a graph and the amount of energy that is going in the reaction, and this is time, the reaction needs to have a little bit of energy to go in. This is called the activation energy. So with an enzyme, the activation energy level is going to be lowered. Here, we can see that the amount of energy needed to get up over that hill has lowered, so the reaction will actually happen faster. So the green one here is with the enzyme, and this one here, the blue one, is without the enzyme. Enzymes lower that activation energy. They lower the amount of energy you need to get up over the hill to get that reaction started. Let's talk about inhibition. So this is stopping an enzyme from working. Sometimes this is a natural occurrence, and sometimes there is a foreign chemical that comes into the body that can also cause inhibition, and that foreign chemical can be on purpose. You're taking a medicine or a drug, or it can be a poison that comes into play. So we have two types of inhibition. We have competitive and non-competitive. So let's first talk about competitive inhibition. In competitive inhibition, the competitor binds to the active site and blocks the enzyme from working. So the competitor molecule has a very similar shape to the substrate and then can bind to the active site, blocking the active site from the substrate from binding, and thus the reaction won't happen. There is reversible competitive inhibition, where that competitor can unbind from the active site and the enzyme can then be reused. Then there's irreversible competitive inhibition, where when the competitor binds to the enzyme, it can't unbind and is completely blocked that enzyme from ever working again. Nerve gas is this case, where it is a competitive inhibitor that irreversibly binds to the enzymes and then basically shuts down the function of the nerves. Another example of a competitive inhibitor is Relenza. Relenza is a medication used to treat influenza. It was designed by an Australian scientist, and what it does is it binds to one of the key enzymes that the virus utilizes to invade your cells. So one of the binding sites that the influenza binds to get into your cells is blocked by this competitive inhibitor, valenza because it's a very similar shape to that of the virus binding site, and it keeps the virus from going into your cells, thus keeping you from getting sick with the flu, or even more sick, having the flu spread throughout the body. Non-competitive inhibition is when the competitor binds to a site other than the active site. It binds to what we call the allosteric site. If this is our enzyme, this being our active site, the active site again is where the substrate binds. The non-competitive inhibitor does not bind to that active site. It binds to another spot somewhere else on the enzyme, and that is the allosteric site. When the competitor binds to this allosteric site, what's going to happen is it's going to change the shape of the enzyme, and when it changes the shape, it's going to mess up that shape of the active site, no longer allowing the substrate to bind to the active site. So remember, when you change shape, you change function. An example of a non-competitive inhibitor would be cyanide. Cyanide interferes with cellular respiration. It binds to an allosteric site on one of the cytochrome enzymes. Cytochrome is part of the electron transport chain of the in the mitochondria of cellular respiration. And so it blocks one of the key enzymes in that mechanism and shuts down the ability to do cellular respiration and make ATP. And you have no more cellular ATP, so therefore things might go bad. Changing the rate of an enzyme. Things that we're looking at: pH, temperature, sometimes salinity, the amount of enzyme that's available, the amount of enzyme that's available, are all going to be things that change the rate of an enzyme. Some of these things, such as pH and temperature, are going to change the rate of the enzyme, one, because of how molecules behave in warmer or colder environments, and then also because a change in pH could lead to denaturation, basically the enzyme or the protein breaking, the bonds undoing, and the protein unraveling. First, let's take a look at temperature. When you have colder temperatures, the molecules slow down their movement, so there's going to be less movement of the molecules, less bumping of the molecules, because essentially the way an enzyme works is it bumps into the substrate, and the substrate and enzyme happen to chemistry and therefore do the work. So if the enzyme never bumps into the substrate, it's not going to react. So as it gets colder, there's going to be less movement of molecules. The movement slows down. There's going to be less bombarding of the substrate and the enzyme. So it's going to slow down the rate of that enzyme. When we get warmer, the molecules speed up. They're going to bombard a little bit more frequently. So the enzyme will work faster to a point. Depending on the enzyme, will depend on its ideal temperature range. The warmer it gets, the faster it's going to go until a point, until it denatures the enzyme, where the heat gets too much for the bonds to hold together and the bonds of the protein start breaking, changing the function, changing the shape, changing function. Now, most of the enzymes in your body work at an ideal temperature of 37 degrees Celsius, 98 degrees Fahrenheit. So when our body temperature goes too low or too high, we're going to start to change the enzyme rate and change how our body functions. And so when we go above or below that temperature, we might start to feel sick because some of our enzymes can no longer work. Next, let's look at pH. Enzymes tend to have an ideal pH that they work best in. The pH of the solution that the enzyme is in can, if the pH gets too high or too low, can disrupt the hydrogen bonds within the protein, again, changing shape, changing function. So enzymes will have an ideal pH range. The enzymes in your stomach are going to want a lower pH. So if you are having an upset stomach, what you might do is take a Tums or a Pepto-Bismol, what that does is it raises the pH of your stomach and starting to deactivate some of the digestive enzymes in your stomach. If we are to look at a graph of enzyme function versus pH or temp, we're generally going to see a bell curve where there's an ideal pH or temperature of where the enzyme will work best. And then as you get away from that average, you'll start to see the enzyme function drop down. The other thing that can change the rate of enzymes would be the amount of enzyme available and the amount of substrate available. If there's a lot of substrate, there's a lot of reaction to happen, and then at some point the amount of substrate available is going to kind of level off, and you won't see much of a speed up. So generally you're going to have this sort of graph where, again, this is the function of the enzyme percent activity, and then this is going to be the enzyme or substrate concentration. You're going to have a sharp increase, and then it's going to start to level off as the enzymes are being used. As the amount of enzyme increases, you're going to have more enzyme to break down. As the amount of substrate increases, you're going to have more molecules to bombard with, until you get to a certain point where you start to run out of substrate to react with, or you start to run out of enzyme available to do the job. Alrighty, that was enzymes. Thanks for watching.